Hi everyone, so my name is Ibrahim. Yes, as you can see, I'm going to give you a similar vision from the eTRIKS standpoint. We obviously heard this morning Brian saying clearly in his presentation, this is not a forum to discuss tranSMART 2.0, and I was just thinking to myself, like, well, this is really the topic up there. I mean, we're talking about a vision and a high-level roadmap, so I just started getting slightly nervous about that. But then in the midst of his words, he said again, it's crucial for us to discuss where tranSMART fits with the other projects of his interest, and we really need to understand it. Like, yes, so that comes in what we're really trying to put here as a proposal of where we might see tranSMART fitting in. So we've also heard about this sort of top-bottom and bottom-up approaches, and so I just wanted to start off with those very my popular sort of quotes there, and this sort of presents to us what this top-bottom is. And we always have this idea that we want to be able to answer these questions about specific diseases and as well at the same time do something that is more beneficial to patients and stuff like that. So why not do both? And at the same time, we always have to achieve this feedback loop between basic and clinical. So these are all ideas that we always think of as top-bottom, and when we're trying to solve them, we all understand it. We can't deny that we think of these things. But then at the same time, we can always think of working it bottom up, looking at the projects. And this is how I'm going to also present it to you by one of the projects that we support for eTRIKS. So basically, I mean, that's just to put eTRIKS in a very, I mean, you talk about high level, and this is even like the topmost. It's just about saying that eTRIKS is really, it aspires to be a commons framework. It aspires to become a platform, an open platform for all the initiatives for in translational medicine, and obviously enabling scientific knowledge with all the basic research, and as well present means to provide new approaches and new tools for prevention diagnosis. So just again, because we've touched on that this morning, I think in Kay's presentation, this is again just to put eTRIKS in the scope. We're not really trying to produce that one framework that will solve it for everyone. It's not going to provide a one solution for all of them, but it tries to establish this commons infrastructure that then will enable the other projects to build and expand and share the contributions towards such an infrastructure. And that's why we are still very much forward to that core infrastructure and the open access bits. So as I said, this is looking at it from a more project-driven approach. So we're taking it as one of the IMF projects that eTRIKS supports, which is viewed by PRED, and it stands for the Unbiased Biomarkers Prediction of Respiratory Disease Outcomes. Generally, I will not go too much into that, but it's just taking it as an example. That the aims of it is to improve understanding of severe asthma and be able to use individual characteristics when they prescribe later on treatments towards personalized medicine, all that stuff, and obviously trying to overcome bottlenecks in the whole pipeline of translational medicine, which seems to be a common aim for most of these projects within that realm, right? So briefly, this is sort of the methodology of this project. They start by creating cohorts of asthma patients and subgrouping them by asthma subtypes, then do all the different omics or clinical profiling and molecular profiling that they can do about these patients. And the aim is to generate these handprints. So basically translate the data into what could become a handprint to use it as a phenotype handprint against these different asthma subtypes, given the analysis from clinical data, integrating the clinical data, the metabolomics profile, the proteomics, transcriptomics, and the patient-reported outcomes, put this all together in some sort of a systems biology approach, and be able to refine those again in their models. They work on devising refining these against animal models and in vitro experiments. So this is their general workflow. They refine, they validate, and then obviously what they want to come out at the end is outcomes about disease models, predictive biomarkers, clinical models, and targets for intervention. Okay, next please. Right. So if we yeah. Okay. Oops. Where does it go? So just looking at the same workflow, but from a perspective of what eTRIKS as a platform could support the management of such a methodology, then you can see that the bit where they create all these studies, they need an infrastructure that supports study management, right? All the clinical studies, all the different design types that are included in these clinical studies. They're not just clinical trials. It could be they have cross-section, they have longitudinal and different design types, and also all the myriads of different omics studies that everyone is aware of. Then you get to the analysis where they do the core discoveries, the experiment, exploratory analysis, and all the analysis bit that you need. Then you get the results. You want to create this integration of all these different profiles and map them to these handprints and validate them against external resources, check other data, prior knowledge, and then obviously go back into the same loop. So just looking at these main points, these are sort of what we can start to see as the main components of what an infrastructure that supports these projects should support. So as just taking this as an example, as UiPath is being supported by eTRIKS, this is just a use case of what one of the queries that users in UiPath want to do is basically to compare animal models and humans just to get gene signatures that are comparable with respect to certain phenotypes. And then they would use tranSMART to, via the gene expression signatures, be able to find these reciprocal models between the animal and the cell models. So obviously you get, again, we're going back to study management. So we have somewhere where you define your study objectives, you record your research hypothesis, you design and you document your analysis workflow. Then somewhere where you actually, this is where tranSMART comes in, as well as in the analysis part where you can do your subcohort selection, then you do your statistics, you can then look at the gene expression signatures, be able to export them. So this is all the analysis bit. And then obviously you can also want to plug in with third-party tools. So the idea is that you're not just running the in-house sort of built-in tools, but you want to be able to expose these to other third parties, tools that you want to get your data to or use. For instance, in this case, using Cytoscape to do the gene set enrichment analysis. So, and then obviously once you do your analysis, you come up with these interpret results. This is where your added knowledge comes in, right? At this stage, you've got your report where you put these results in it. You're recording your study findings, and then you're also providing in a sort of a notebook, in this case, format, your provenance of how did I achieve this and what is the process that I've been through. So you need to have somewhere of a provenance support to. In this case, it's more of a process provenance that shows how did you arrive at these outcomes. So looking through these different steps and these different components, you can start to think of eTRIKS as a platform that needs to deliver these sort of list of requirements, right? That is sort of, they're not really features, but they're just an infrastructure for what other features could then start to build up on top of those. So you obviously need a repository for the study data, including the omics and the clinical data set. Then you need an open access platform. And in that case, we're talking about, again, this clear distinction between how you expose your data and you provide open access and analytical tools to your data. And then we've seen how you always, like in this case, about these handprints. This is an integration step. This is something that doesn't exist in any system, right? I mean, it always end up in papers or in publications. But if you can actually capture how these scientists put these interpreted results together and produce these handprints and be able to store them in a more of a knowledge representation. And I think in one of the presentations there was this idea of knowledge representation in a repository, so that you can be able to grab this information back and feed it back into your new studies and build upon that. And obviously within that we just spoke about one IMR project, but the idea is that because this is meant to support different projects, then you expect that it needs to be a collaboration and provide this collaboration environment where multiple researchers share their input. And then, last but not least, it's the idea of supporting the scientific evidence, the provenance, supporting all the analysis workflow provenance and the multiple usage, right? So in a way, that's just a proposal and an attempt to start looking of where these things all can be put together. So And I think it just presents itself really. It's just those main components again. So we have a component that represents the study storing the data in a study-centric way. So as the data comes in, you want to support the data, the metadata about the study, and obviously you have then exposed this via an analytical commons, where basically you allow users to build their own tools, and you expose it via APIs that people can then start. You obviously provide your own collection of tools, but then you want to make it easily accessible that you can build tools against it. Then you have this other sort of separated component here, where you're storing your master data, stuff that tranSMART sort of pulls in all together. We have the master data or the research and documents, and we can't get away without that, but we need to make it clear that these two things could live in two different worlds. This could support the study, but that could support the results, right? And the results live in the same world as the external resources that you bring in, or the information about the diseases and the compounds. You can then have a different representation of these results in that different repository. And obviously, underneath all of that, you want to expose that via a collaborative UI infrastructure. So you've seen here that I've actually put tranSMART core in this bit, and that's what we see tranSMART starting within that infrastructure. So it starts as the component that supports the study data management, right? And then exposing the data in there to the analysis point. And that's just summing up what I've just said, these four components. And obviously, it's because I'm talking at a very high level here, it's implicitly required that we all have these services that we need to implement: data methodology, data loading, curation pipelines, and problems. Okay, so that's just to, this is getting now closer to what we see as a sort of a high-level roadmap. So as I said, this tranSMART being the tranSMART core that we're trying to work towards now becomes the study repository for translation research. As it is now, this is what tranSMART supports in the database: clinical trials, microRNA experiments, gene expression profiles, biomarkers, literature, and documents, all in this big schema. Although they have the app and search app, but nevertheless, they're really just all ad hoc added tables that people keep on adding and adding without really any common understanding in it. So what would be a fine line here is to define what the core of tranSMART core DB, I'm talking about DB here, is the studies. So anything that relates to the study, the clinical trials, the microRNA experiments, and obviously we need to make it more generic to accept other designs of studies. And anything that related to the supporting information or the results becomes outside the tranSMART core, meaning the biomarkers and the literature and the gene expression profiles. So that's regarding the analysis. As I said, that also becomes externalized in a different component on its own via the same APIs plugin architecture. These are the tools that we all know that the current tranSMART implements. But really, what we aspire to achieve is having this tranSMART core and exposing a clearly defined data API against it. We can start building service APIs that maintain the functionality of what the application wants to achieve, and then expose these different tools. We can have an R API for the data, JavaScript, whatever, and people can start building their own scripts, and you can then start pulling in, pretty much like in Galaxy, you can start having multiple scripts that do different, build up your own workflow process, and this is where you can start also instantiating workflow templates and saving them. And because eTRIKS is also providing a hosted infrastructure, there will be, again, Keith mentioned that the R Cloud can be externalized running on its own R server, R Cloud, where the results it's running on that independently of the application. And all the user gets is he's got this, we call it out, give it any name now, this analytics dashboard where they come in, they pick the studies they want to start working against, they put the processes and they build the tools that they want to achieve and run it on the R Cloud, and then they get the results. And that's just, again, a simple representation of what this knowledge base. It's really what the data is about. So if you think of the tranSMART repository as it stores what the data is like, it's the high dimensional data, it's the clinical profile. What this is that it actually now thinking of this is how we get the data, right? We have a patient or we have a sample, we have these profiles, and these profiles could be mapped to the disease models or the relationships between these five results. And then typically these handprints find a way. And I'm just saying now it's not a particular set in stone, but I guess everyone will get the idea of how we can, why we need that. So as a high-level roadmap for Q3, and as it was suggested on the agenda, current tranSMART database, and we're just talking now database, is got those three main schemas, right? The BioMart, SearchMart, I2B2, and the app. And they're really, and I've got this from one of the PDFs that described them, and they're capturing subsets that are tailored to specific application needs. And that's why we have a dedicated schema for what is the Dataset Explorer. Dataset Explorer is a tool. And we have a whole dedicated I2B2 schema for that tool. That's what we're supposed to do. So I guess what we aim to start working towards, I mean, obviously, we've started working towards the tranSMART app API. We also should start working towards a tranSMART DB, thinking about decomposing all these schemas, separate the management of the study from the other tool-specific data and the master data that we have all in the same schemas, right? So by defining and decomposing into what is the study metadata model, what is the clinical data model, and what is the omics data model. And I guess traits even have a ready omics data model that seems to be generic and distinct from the others. So that's my final slide. That's just an approach of how we can approach thinking about the data model for the tranSMART being around these three main concepts, right? So we establish a study, we establish the concept of a study because we need to be able to do cross studies afterwards and be able to being your core object to store all your metadata. And for the world of the clinical data model, that's represented by a patient. And then you can build up on top of that all the other stuff that relates to clinical worlds. And in the molecular profiling or in the in vitro model experiments, there's a debate around assay or sample, but let's just call it assay or sample in this case. But that is then becomes your core object in this world, right? And that's just dotted lines because sometimes you will have samples that are driven from patients, and sometimes you just have samples, like in the case of in vitro samples. So in this case, you're not really restricted to define a study as a clinical study with patients, but you can define a study by just a sample set. So it's just working towards such a very high level model and then expanding on it, obviously,